So yesterday we talked about the fact that we follow 6 steps when we are developing and enacting a NENS project. And these steps are done in accordance with international best practice. They're not things that Michael and I have just made up. There's some substance in behind these. And yesterday we talked about how we decide if a project is feasible and the considerable amount of homework that we need to do before we decide whether it's appropriate and suitable to embark on a NENS project. We then talked about if we have decided, yes, it is something that we should pursue and we have the funding, we need to do surveys. We need to do surveys in your countries to check what weeds are present, that we have got them correctly identified, see if there are any natural enemies already there. And what we usually find when we do these surveys is there are things there that you probably don't know about. Michael's a master at finding lantana insects that aren't recorded as being present in any country. And that's because these things are moving around, some of them on wind currents through the Pacific. And so we need to check that a natural enemy is not already present before we go through a process of looking to import it. And we need to see if there's going to be things like predatory ants or spiders or wasps that might disrupt the natural enemies. Then sometimes we're lucky and we already know what the natural enemies are that we want to release to control a weed. But other times we're working on projects which are completely new, where nobody has ever looked at that before anywhere in the world. And we have to do surveys in the native range, where the weed comes from, where it originated, to find out what our natural enemies are that we could consider and test and decide if they would be suitable. So today we're going to talk about the next 2 steps. How do we select suitable natural enemies? How do we know they're safe? Safety is the big question. It is the big thing in the front of our minds. And your homework is going to be practicing how you explain to people when they ask you and they think this sounds dangerous and dodgy, how you're going to explain that this is a very safe process. And then we're going to talk about getting permission to introduce natural enemies to your countries and how we go about that. So first of all, something that most of the world doesn't know, most people, is that what we call host specificity is common in insects and pathogens. So that means most insects and most plant diseases are specialized on one host or a group of closely related plants. Most insects and diseases in the world can't attack every single plant or lots of different plants. And an example, I've got a picture here, and I'm not sure how many of you are familiar, but this is a very popular thing to do in New Zealand, to get a monarch butterfly, which is a very pretty butterfly, and to grow the caterpillars on and watch them develop. And children really enjoy studying nature by having some monarch butterflies develop in their back garden. But there's one problem that happens a lot. The caterpillars start off really small and don't eat much when they're little, but they start to double in size and they very quickly strip the plant. The host plant is called a swan plant. And next thing you know, the caterpillars have run out of food. And if you can't find another plant, if you can't go to the shop and buy another swan plant from the garden centre, then your caterpillars starve because they can't actually feed on anything else. They can only feed on the swan plant. So that is a really good example of the kind of specialist type of natural enemy that we are interested in when we're doing this work. We are not interested in working with generalist species, and they are the things which have caused all kinds of problems through being released in the past, the things like the African giant snails, and we are distancing ourselves from things that were done with generous species that were very unwise and have caused a lot of harm. Another example is this picture here, which is lantana, and that is affected by a leaf rust. This is a rust that we have released here on lantana in New Zealand. Now, what we refer to as lantana is actually quite a few different genotypes, and you'll notice some of the lantana has different coloured flowers. The lantana leaf rust will only attack some of those genotypes. So it particularly likes the yellow and pink flowers, but not even all of those. It is so specific, it won't even attack all of the kinds of lantana. So sometimes when we are doing this work, we find natural enemies that are actually almost too specific, and you would have to find different strains of this rust to attack all the forms of lantana that are present. Now, how this works is that we talked yesterday about when we're doing NENS work, we need to understand how plants are related to each other, because the relationship that they have, if they are closely related, means that they have similar chemicals they use for defense. Because, as you know, it's war out there, and the plants don't want to get eaten up by the insects. They're trying to protect themselves. And so they have chemicals in them to try and deter insects from feeding on them. So in order to be able to feed on a plant and use the chemical and not be poisoned by it, you have to be specialized, and you have to have evolved with that plant and have the correct enzymes, for example, in your digestive system, that you can use that chemical safely and develop on that plant and not be poisoned by it. And to go down that particular strategy is means you're very closely tied to your host, and it's not easy for you to be able to change. A lot of changes would need to be made, and we'll talk about that a bit more soon. So most insects and pathogens in the world can only attack either a single host or groups of closely related plants. We have to do testing when we are looking at NENS to be confident and to be sure that we are working with just the specialists. And again, we have internationally accepted guidelines that we follow very carefully to make sure the testing that we have done will give the correct answers. And there's a bit of an art to this, and it's the sort of thing we would get peer reviewed to make sure that the testing we're doing is appropriate and would stand up, international scrutiny. And we are using a process we call a centrifugal approach. I don't know if any of you have seen a centrifuge. It's basically a piece of lab equipment which is round and it spins things in a circle. But what we mean by this is that we start with the host plant in the middle, and then we find out which plants are closely related, and then which plants are more distantly related, and then which plants are getting really quite a long way away in terms of relationship. We need to understand that, and then we can test the plants, first of all, that are most closely related to the target weed. They are the ones that are most likely to be attacked. If we are working with a natural enemy and it cannot attack the most closely related plants, then it's very, very unlikely that anything more distantly related will be attacked, but we will do the testing to be sure that we understand what the host range is and that it is acceptable. So some of these tests can be actually quite brutal. We do what we call no choice or starvation. So we might do something like put an insect on a closely related plant to our weed and say, eat this or die. So when you do such an extreme test as that, sometimes the insects are desperate and they will try to develop. And then you'll quickly find, no, they're trying to feed, but they can't. they're being poisoned by the chemicals in the leaves. They can't survive on it, and they quickly die. And so what this does is it gives you sort of an overestimate of risk, really. In the field, in a natural situation, they would never actually try and attack those plants, but because we're forcing them, they are trying to. And so that's it gives us, you know, the worst case scenario, which we know is not going to happen in the field. But it's often more appropriate to do what we call a choice, because this is what happens in real life. Insects have to make choices, and they have all their senses. They can smell volatiles from the plant, they can taste, they can see, and so they make choices about which plants they go to and land on, which plants they feed on, lay eggs. So the word oviposit there means to lay eggs. So it can be more appropriate to give them a choice. And what we usually find in a choice test is they go onto the host and they ignore all the other plants. And that will show us, you know, what will most likely happen in the field. And to do the testing, we have to figure out for each agent what is the most appropriate thing. So we have tests which may just be this oviposition or egg laying. If an adult will never lay eggs on another plant, there's no need to test really whether a larva could feed on that plant, because the eggs will never be laid on it. But often what we will need to do is to test, can they actually develop? So if sometimes, particularly when we're working with moth species and doing testing, the moths go a bit crazy and they'll lay eggs on everything, including the cages. And then we need to see, well, what happens? They're getting confused, they're laying eggs. If that happens, will those eggs hatch into healthy larvae and will they develop? And usually they don't. And we can do tests either in containers or cages, or sometimes it's best to do them in the field. And what the problem can be when we are doing testing inside containers or cages is that the insects get confused. The plant volatiles are getting all mixed up. So their host plant in a small room with other plants, they can't distinguish the host plant. They're getting confused because the smells are all mixed up. And in those sorts of situations, sometimes we need to do field tests under much more natural conditions. Testing is not usually done in country unless you have a specialised containment facility that you can do the testing in. Now, these specialised containment facilities are pretty few and far between. We have a couple in New Zealand and there are a couple in Australia. There's one in Hawaii, but otherwise they're not available. So we are having to use these facilities for the Pacific projects so we can do the testing and be sure before a natural enemy would ever come into your country. So we are happy to help with that work, and it's, you know, these containment facilities cost millions of dollars to build. So it's probably likely that we will need to help you with these aspects of the work. Testing can give what we call false positives. So what I mean there is these situations where the testing is a slightly artificial environment and the insects are desperate because we're giving them no choice, or we're confusing them by putting them in a small cage and they're trying really hard to attack something. But the other point I really want to make here is that when we test natural enemies, we do reject some. Sometimes I think there's a perception out there that, you know, we are crazy scientists who will stop at nothing and we'll be determined to pursue a natural enemy even if it's not safe. But that couldn't be further from the truth. If we don't think a natural enemy is safe, or if there's even any doubt, we don't proceed. And this picture here is an example of one that we tried to bring in for woolly nightshade. Some of you have that plant in the Pacific. It goes by a range of names. The Latin name is Solanum mauritianum. It's a poisonous small tree. We spent quite a lot of work on this weevil to attack the flowers, but it was going to attack one of our native Solanum plants called Poro. And so we walked away from it. And we get a lot of credit by sharing the stories of the natural enemies we have tested and found unsuitable. People like to know that we are rejecting some, and that if in doubt, we don't proceed. We have a bit of an advantage here in that we discussed on the first session. There's actually a lot of natural enemies that are already in the Pacific, that are in some countries but not in others, that could be shared more widely. And these are very well known natural enemies. There's been a lot of testing that's already done. Where there is interest in introducing them into a country where they're not already present, we do an assessment to see whether there's any additional testing that's required. Does that country have native plants that are unique to their country that haven't been tested, or maybe economically important or ornamental species that we need to check if they're at risk of attack? But often there's little to no extra testing that's required because the flora is often quite similar between your countries, and we already have enough information that we know these natural enemies are very host specific and will be safe to introduce. And by using these tried and proven natural enemies, we can reduce the cost of projects and increase our chances of success. So it's much easier working with things that are well known or already in the Pacific, but that doesn't mean we don't need to tackle some of the harder things, because there are other weeds needing control, and we do need to develop natural enemies for those, and we are working on that as we speak for a number of new targets for the Pacific, which we've touched on, and we will touch on a bit more too in session 5. Now, this is a question you can guarantee that you will get asked. What will happen if your natural enemy runs out of food? Well, natural enemies hardly ever eradicate their host plant, and only usually if they do on a local scale, but not from the whole country. Because as the plant becomes rarer, it's harder and harder for them to find them. And some escape attack because they're just harder to find, or they're hiding somewhere under another plant. For example, the natural enemy doesn't find every single host. And because this is a natural system, the natural enemy doesn't want to kill all of its host and have nothing to live on. So nature doesn't work that way. Instead, if we are successful, the host plant becomes less abundant. So there's less food, there's less habitat for the natural enemy, and that population declines as well. They're not able to jump off and eat other plants. And when we are successful, a weed and its natural enemies can become quite rare. So what you will see, instead of a paddock full of broomweed, you will still be able to go and find one plant in that paddock, in that field, and on that plant will be a couple of broomweed beetles. There won't be thousands, because that plant can't support thousands. There will only be a couple. But if something happens and the weed gets bad again, now we know a lot of weeds have seed banks. So they make seeds, and the seeds go into the soil, and they can live for a long time in the soil. I know there are weeds here in New Zealand that can live for 50 or 100 years as seeds in the soil. And so if the seeds get a chance to come back to the surface and to germinate, the weed can come back after being absent for a long time. And if you have something like a cyclone that comes along and removes vegetation and stirs things up, you might suddenly get a resurgence of a weed that hasn't been so abundant. Because there's lots of food, the natural enemy can then build up again in number. Excuse me, I'll just have a sip of water. So those two beetles that were sitting around on that broomweed plant can breed and become 200 and 2,000 and 200,000 and so on until they again get on top of that weed and bring the abundance back down. So it's not unusual to sometimes think, oh no, the project's failing. The project was working, but it's failing. The weed is back. And we have to have patience and trust that the natural enemies, they're still there. They will catch up and they will do the job again. So this is how we often see a project proceed. So you start off here with this blue line. That is the weed, a high density of the weed. And we're starting off down here by releasing a natural enemy. And for a start, there aren't very many. We might only be releasing 50 beetles. And it takes them a while to build up their numbers and to get to the size of a population that will reduce the weed. And down comes the weed, and down comes the number of beetles. And then the weed maybe tries to have a bit of a resurgence, and the insect catches up again. And so on, this happens over years, decades, but the general trajectory is downhill. So the ability for weeds to bounce back becomes reduced the longer the project goes on. The seed bank gets used up and the weed will become increasingly rare. Another question we often get asked about is evolution. And people get really worried there's going to be mutations. So, you know, you've told us this natural enemy is safe, but how do you know it's not going to go up and mutate and turn into something else? So from the 100 years of information that we have about the use of natural enemies all throughout the world and in the Pacific, we know there is no evidence to date that any natural enemy that's been released to control a weed has evolved to attack any other plants. So that's quite comforting. We have 100 years of data and experience. And the thing we have to understand about random mutations, yes, they can happen, but for a random mutation to be successful, it needs to be beneficial to that organism. And quite often they're not. Living things are very complex, and for one gene to change and for that not to then harm other parts of the way that organism functions doesn't happen very often. And even if it's beneficial, there's substantial barriers to overcome for that to persist and to perpetuate and to become common in a population. Because basically that's just one individual who's got that random mutation and all the other individuals don't have it. And so the way that the genetics works is that that just gets swamped and it disappears. So this is the reason why it's actually very difficult for these natural enemies to change, which isn't to say that it's not impossible in the longer term. And we're talking about thousands of years. And the risk of any of your native insects going on to new hosts is the same because it's the same mechanism. And it's been calculated that the chance of a host switch happening is between 1 in 10 million and 1 in 100 million. So these are very, very small numbers, very tiny, tiny risk. But on the subject of evolution, we have done some work, and including some work here in New Zealand, that there can be some evolution just to become better adapted to climate. So we had a beetle we released here to attack a plant called heather, which is the one they love in Scotland with the purple flowers. And we released it in a part of New Zealand where we can have a range in temperature from 30 degrees during the day to -10 overnight. And the beetles, even though they came from Scotland, had never encountered anything like that, because it's just cold in Scotland. It never gets that hot. And so only the big beetles that had lots of fat reserves were able to cope with those conditions and survive. And they have then gone on to breed a strain of them that can cope with the conditions in New Zealand. But that has taken several decades for that to happen. So we can get evolution to get better adapted to climate, but the chances of random mutation and switching on to other hosts are perishingly small and not something to lose any sleep about. And again, we need to put this into perspective. There is nothing in life that is without risk. Every time you get in a car, you're taking quite a risk. Even you can die from drinking too much water. There's no life without risk. And what we know is that if we do nothing about invasive weeds and other invasive species, we're having great harm done to our ecosystems, the services they provide, to your biodiversity, to the livelihoods of your people, their well-being, and animal welfare. We know that the safest and the likely most effective way of controlling widespread weeds is by using the natural enemies. And when we do this in accordance with the best practice, which we totally promote best practice, we're not interested in doing anything other than best practice. It is very safe. We should be much more concerned about new species which are accidentally invading our islands every year, things that are being smuggled in. I've been at an airport, I won't name the country, and we know what's coming in people's bags that they are not declaring on their card. And also new plants and crops. We all need new plants and crops to grow, but we need to be very careful that they're not going to go on and become just yet another weed for us to deal with. So it's all the things that are coming in that aren't being tested, and all the things that you've got already in your countries that we know are a problem that we need to be worrying about. The natural enemies are thoroughly tested and studied, and we only proceed if they're safe. Right, that was quite a lot from me. There was quite a bit to tell you then. So we're going to go back to Mentimeter, and we're going to get you to rate now from 0 to 5. How comfortable, having heard what I had to say, are you in the use of natural enemies for weeds and that being safe? So if you're still not at all comfortable, you might be 0. And if you're very comfortable, scoring a 5. So we're dropping. We were up to 4. Oh, back up to 3.8. Okay, so we're doing, oh, no, we're back down to 3.7. Oh, 3.5. No. Okay, so those of you who are not sure, can you please tell us why? I'm interested to know why you're still concerned. So we haven't had any responses yet. It would be great if you could tell us, if you maybe scored less than 3, why you're still worried. You are completely anonymous. It would be helpful to understand. Thank you. Kind of equipment we use here is not good enough. Okay, I can actually say something about that. If you're working with us on a NENS project, we make sure you have adequate equipment as part of the project. And we would not advise introducing a natural enemy until testing had been done elsewhere with adequate equipment. Lack of certainty in the Pacific. I'm not quite sure what that one means. Perhaps you could provide some more detail for us there. Is that because you think there's not enough information about natural enemies that have already been released in the Pacific? I'm not quite sure what that one is. Lack of awareness in our community. We talked quite a lot yesterday about we have a big piece of work to do if we're going to work on natural enemies, to bring our communities along with us and to talk to them early. And every team who's going to get involved in NENS is going to have to do quite a lot of raising awareness and having discussions and talking to the community about the worries. Malo, thank you. Because not all native plants and animals are known. I think that's true for microorganisms, but I think we have a pretty good handle on what native plants are present in the Pacific. Maybe some more work to be done, but it's amazing the databases which are available, and perhaps Michael can talk a bit more about this. He's been doing a lot of study finding out what is known about what plants are where in the Pacific. Danger to play with nature. Yes, I agree, it is dangerous to play with nature if you don't know what you're doing. May disrupt food chain.
I think the thing to bear in mind with that comment is that we're not actually playing with nature. What we're doing is we're introducing a natural enemy into an environment where it's absent. And so we're not actually playing with it, we're actually restoring the balance of nature. With it, when we have weeds without control, they are actually disrupting nature. And what we're trying to do is put a balance back in.
Yep. And then there's a cluster around the lack of knowledge and limited knowledge. So that's one that we can relatively easily address by making information available. And, you know, while we're making these sessions available on YouTube and providing resources, there's possibly also work to be done about making materials in other languages and in terminology that people can understand. So there's still, yeah, quite a bit of work for us to do. Very interesting, the testing process seems to be a long process and lack of skilled professionals in country. Yes, the testing can be a long process. We have had some projects where it's taken 5 years before we were convinced the natural enemy was safe. And for a lot of countries, you will need to rely on people who do have the skilled professionals to do this work, because it does take years to train to be a scientist to do this work. And yeah, we will need to use the people who are skilled to help. And then I'm just scrolling down a bit. May conflict with traditional views. Well, that is definitely part of the conversation to be had very early on with communities. And I think it's always really important to talk about, well, why do we need to do something about these weeds? What's going to happen if we don't do something about these weeds? What's at risk and what are you going to lose? Another one here, not sure which natural enemies to use on weeds in our country as a control. So that's something we can definitely help with. Michael's developing some very good information resources around what's known to be present in the Pacific and in which countries and what the opportunities are. And need to familiarise with weeds here and how to prioritise them. Yes, there's a, you know, it's quite a skill to be able to identify plants, and that's maybe something we need to think a bit more about, is how we can provide support to train people how to identify plants and other natural enemies. And we're going to talk about prioritization in session 5. So thanks for providing all that useful context. Are there any questions that you would still like to ask about safety that you would like Michael and myself to answer right now? If you have a question, please submit it. All questions are good as far as I'm concerned. And if people are typing in questions, you can like them so they come up first. I'll just wait another minute to see if any questions pop up. Okay, well, we must have, good, thank you. Question. Any examples of failed weed biological control? And yes, there are. And Michael mentioned this the other day. Michael, do you want to just answer that question again?
Yeah, so there's been a number of projects where biocontrol hasn't worked as much as we would like. This doesn't mean to say that the agents have attacked anything else. It just means that they didn't control the weed on which they were released. And so a classic example in the Pacific was nutgrass or Cypress rotundus, where there's been a number of biocontrol agents released, but they haven't really been effective. They're out there, they're feeding on the plant, but they're just not controlling it. And whether we need to look at more agents, I mean, that's another question. Depends on how important that weed is. With lantana, we mentioned lantana a fair bit. There's lots of biocontrol agents. There's about 24 released globally on lantana, but there's probably only about half a dozen of these biocontrol agents which are actually damaging to the weed. And so a lot of the insects which are already out there on lantana, they're not attacking anything else, but they're not controlling the weed. They're not aiding control. And so they're the ones that we don't recommend to move to other countries because it's just a waste of time, because they're not going to do well in one country, they're not going to do well in another. So we only recommend agents which are not only host specific, but also agents which are effective. So we don't want to give people dud agents in that regard. So there are a few examples, but Lindy said in the very first day, 60% of our projects are either successful or partially successful in biocontrol. So we've had a very good strike rate.
Thanks, Mike. Yes, so we've got examples in New Zealand too where we have failed. Sometimes we're not able to rear the natural enemies and release enough of them to get them established. But, you know, so there's some room for improvement in terms of picking the best agents that are most successful and making sure we can establish them. But the failure is not around host specificity. If natural enemies are mutated, can it affect other non-target organisms? Now, as I explained, if a mutation arises, it doesn't persist. So it's just one individual will get a mutation and that will die with that individual. It won't be passed on.
I think it's probably worth mentioning a little bit more about mutations, because it comes up a lot, and so does evolution.
Yeah, thanks, Mike.
But if you've got an insect in the field, whether it's a biocontrol agent or not, when it goes around looking for its host, first of all, it smells the plant because there's a sea of green out there. And then it gets closer to that plant, it's got to be able to then see the plant. And then an insect would land on the plant and touch it with its feet. It's got taste receptors in its feet. So that's 3 things that needs to change already in host finding behavior. It's got to be adapted to the right smell of the plant. It's got to be able to see the plant, and it's got to be able to taste the plant with its feet. And then if everything else is going fine, it'll take a sample bite. So it's got to be able to process it. And then the insect needs to have all those enzymes in its body to be able to utilize the plant chemicals for it to be able to be processed. And if it doesn't have those those enzymes in the body, it can't process the plant. And so for a mutation to occur, if only one mutation occurs, then there's 4 other or 5 other aspects of host finding and utilization that the insect also needs to do. And the chance of having 5 mutations simultaneously is close to 0. And the other point I want to make about evolution is this, is that evolution actually works to make insects more specialized, or any animal more specialized. So think about this. If you had an insect which is a generalist and it eats a lot of plants, it's got to have all those resources and all those senses to be able to recognize all those plants. But it also needs to have all those enzymes to be able to process those plants. And producing enzymes to be able to process plant material is a huge physiological cost. And so it's not in the interest of the insect to have all these enzymes, which they're not going to necessarily use. And so an insect then evolves to become a more specialist, because then it only has to produce 1 or 2 enzymes and not 10 or 20. And so evolution actually works to make things more specialized. And this is why some of the biocontrol agents that we utilize are just attack the target weed. In fact, when we go back to the country of origin, we only collect these things off the target weed. We're not collecting them off any other plant. We're just collecting things, insects or diseases, which are only found on those particular plants. And so evolution works in our favor, and just the fact that mutations also work in our favor, because an insect, the chance of 6 mutations occurring simultaneously is highly remote. And so I think, we need to look at those things in the big picture. I hope that sort of helps.
Thanks, Mike. And yes, we will find some papers and some information and put it up on the shared drive for you to access an answer to this question here. Is there a standard screening process in place when introducing a new organism? We are going to talk about the steps beyond the testing, which we've just described, and the sort of import risk assessment. So we're going to talk about that in a minute. So hold fire for that one. Is that a good idea to introduce natural enemies from other countries? Well, I would say yes to that. We have done it for 100 years very safely and successfully. If we follow international best practice, it is a good idea, but it does need to be done carefully and properly. If this technique is safe, why has it not been readily used in the region before? Well, Michael gave some statistics in the first session about actually how many countries in the Pacific have done this. Have you got those at your fingertips, Michael?
Yeah. So using natural enemies against weeds has been in place for over 100 years. Fiji was the first country after Hawaii to introduce natural enemies, and that was done in 1911. And so 17 countries out of the 22 picks have deliberately introduced a weed natural enemy to control a target weed. So that's almost all of them, except for 5. I think the problem is that many countries haven't released a natural enemy against a weed for 10 or 20 years. And so it's forgotten about. They've forgotten that it ever occurred. And this comes up in workshops from time to time. But 17 countries in the Pacific have already introduced agents. And at the moment, in the last 5 years, I think 5 countries, PNG, Vanuatu, Cook Islands, have all introduced natural enemies. So maybe it's something that we need to get more, make people more aware of what's happened. And there is a paper on Google Drive which talks about the status of biocontrol in the Pacific. You can have a look through that.
Yeah, we refer to it sometimes as the forgotten tool, because when we are successful, there is nothing to see, which is slightly unusual and ironic thing. So if we're successful, there's no weeds to see much anymore, and people forget about them. An example of a biocontrol agent that's been a vector of plant disease. I'm not aware of any. Are you, Michael?
There's a couple of insects that are vectors of plant disease. Some of the tingids have been. However, what's important is that since the insect only feeds on its target plant, it's moving the disease of that plant to another plant of the same species. So there's an insect on Lantana where it transmits the disease in saliva, and then it helps control or impact on that plant. So it is out there. It's not common with a lot of insects, not the biting and chewing insects, but the sap-sucking insects certainly can transmit disease. But please bear in mind that the disease, which is in Lantana, is going to get transmitted to another Lantana plant, because the disease is also host-specific and it doesn't attack other species because it is also co-evolved with its host. And so, in actual fact, the biocontrol agent is being useful by moving this disease around from host plant to host plant.
Thanks, Mike. And I know this is the reason we don't work with aphids, for example, as natural enemies. And it is part of the risk assessment we do. If we're looking at natural enemies, are they likely to be the type of insect that might have a virus or something that could be transmitted? And we would probably not proceed with those unless we could show they were safe. So I know there's ones we have walked away from here because we thought there might be some risk. So that is definitely considered and as part of the risk assessment. Have there been cases where natural enemies go out of control, affecting more than what they're supposed to? The only cases of this happening are where decisions have been made to release, like for example in the United States, they had thistles, which are very prickly, nasty little pasture weeds that were really serious, and the farmers were unable to farm with these thistles. They also have native thistles, but they didn't care back in the 60s and 70s about the native thistles, and they went ahead and released some natural enemies to control the pasture ones. And they did move on and attack the native thistles, but that wasn't a failure of the testing or the risk assessment. That was a failure of human judgment. And, you know, we would never promote doing that kind of thing. You have to be and we're going to talk about this more in the next bit that Michael's going to discuss around making a decision to release something in country. We need to be very careful and make sure we're doing the right thing. And people have made poor choices in the past around introducing particularly the generalist natural enemies, the cane toads, the giant African snails, the minor birds, all those sorts of things that has given natural enemies a bad name, but we are not doing that type of work. The weed natural enemies is a very safe and successful process.
It's pretty worth mentioning because the cane toad gets mentioned a few times, but in Australia, the authorities or our regulators actually knew that the cane toad was a generalist and would attack other things. And they made the decision to release, not the scientists. Or the scientists said was, no, this is not a good thing to do. And it was a political decision in Australia, something that they probably regretted for the last 100 years. But as Lindley says, we're not talking about releasing generalists here. We're talking about releasing things which have co-evolved with their target weed, and the testing shows that they are not going to attack other plant species. So I think we need to make that distinction between a co-evolved natural enemy found on the plant versus something which was known to be a generalist, such as the examples that was mentioned on Monday, or sorry, Tuesday, or Monday for some.
Yeah, thanks, Mike. We're going to be talking about monitoring and evaluation tomorrow, and so I'll leave that question for then. Any insect rearing courses available? I'm not aware of any. What we normally have to do is to train in-country teams on insect rearing, or I know my staff who do insect rearing reach out to other people in other parts of the world who are rearing insects for advice. It is, you know, it's a skill like anything else, and yeah, there's all sorts of ways that you can succeed or fail. We're going to talk more about rearing also tomorrow, and some of the things that are involved with that, and some of the things that can definitely make it successful or fail.
I just want to add, 10 or so years ago, Australia used to run these 2-week in-house international courses on biological control, and we had sessions on rearing, we had sessions on testing the insect, plus monitoring and all these things. And people found these quite useful because it was a 2-week course, it was very hands-on. And there's some talks on how to rear biocontrol agents or pitfalls to watch out for, and I think we cover that tomorrow. But certainly there's a lot of information out there which we can supply. And the other thing is that Lynn and I have spoken about is that when we introduce a natural enemy into a new country, it comes with instructions, or it actually comes with a person who is going to walk you through it for the first week or so. And a lot of these insects, by the time a country gets them, they've already been well studied. So we already know how to rear it. We already know its biology and everything else about it. So it makes it much easier for the recipient country to handle it and take over.
I'd also just add there that yesterday we talked about the vision of having a network throughout the Pacific where you guys are all helping each other, and maybe having, you know, visits to come and learn off each other how you've managed to successfully rear insects. I think there's a huge amount of potential value there, and just sharing a bit more widely amongst ourselves. An example or case study where communities were reluctant to accept and get established, and how you overcome this hurdle. I'm very fortunate where I'm working here in New Zealand. Our community is very accepting of the use of natural enemies, but I'm aware that there are other places where that's not so. I know there's been some issues in Australia, and I know there were some real issues in Hawaii around the use of the strawberry guava scale, for example. From my perspective, I think it's about having a really meaningful dialogue with your community and understanding where the concerns are. People need to be listened to. People need proper answers. But also it's about that bigger picture. Do they also understand what's at risk? Do they understand the harm from not controlling the invasive species? Do they understand the harm potentially from using herbicides? And do they understand the risks, you know, the sort of overall cost benefits risks, and are able to make an informed decision? Sometimes it can help, again, to do a tour and go to another country and see that one of your neighbours has done this work safely and successfully, and they are happy, and to ask them, well, what did you need to do to get your community on board? You know, this sort of information sharing. And if you've done all of that, and you've done all of that really well, and your community doesn't want to go there, well, I think you just have to back off and wait until the community is ready. You might have to wait until the weeds have got worse and people are a bit more desperate. The weeds will get worse and people will notice their impacts more. We have to respect our communities and we have to work with them. And this means we have to be very patient and we have to take our time and we have to do twice as much listening sometimes as talking.
I want to give a couple of examples, a good example and a not so good example. We did some work in Timor-Leste and they had never done biocontrol before. So they were a little bit reluctant. All the questions that have been raised in the last 3 days were raised for this project as well. So we've got landholders, we've got communities, we've got departments on board, and all the things that we've discussed get mentioned, including the impacts of the weeds if we don't do anything. And eventually they came around and they embraced it, and we released 2 biocontrol agents, which they were very happy about. But there was another case when I was working on the Mycania rust, and I flew into a country, and I won't mention the country, to introduce the biocontrol agent. And they weren't accepting of it. They were reluctant because there is a perceived view that Mycania is a useful plant and the natural enemy would reduce the plant down to 0 and they won't be able to use it at all for medicinal medicine, traditional medicines. So when we jumped on the plane and flew out of that country, we just said that we weren't going to include it into the project because the communities hadn't embraced it. And that's fine. You know, we can have those talks later on if they change their minds. So not all countries are going to embrace. We accept that, but we'll work with the countries that are willing, and hopefully the more information we get out there, the more other countries realise that it's not so risky and not so scary as they may think.
Thanks, Mike. So thanks for all these great questions that you're sending in, guys. So we'll just keep working through these. We have got enough time to handle all the questions. So how do we protect the natural enemies from destroying the weeds? Well, unless you're actually going to kill the natural enemies, go out there and squish them, they are going to destroy the weed, but that's what we're wanting. The problem can be if they work too fast sometimes and you're wanting to be able to go and harvest them in the field so that you can release them somewhere else. And so, yeah, we'll have to sort of provide a customized advice, I think, there, if that's an issue. When we say biocontrol agents, evolution and being better adapted to climate, is their adaptation being positive or negative? This is positive. So this is about getting better establishment of an agent. The ones that can survive go on and have offspring that are better adapted to the climate. How do we weigh the pros and cons of introducing natural enemies? So Michael's going to talk about this next. So hang in there for a minute or two longer. We have some newly recorded weeds. Shall we start requesting biocontrol even if they're still localized? We talked in the first session, and if you weren't available for the first session, you might like to review it, that we have a hierarchy, I suppose. First of all, we try and keep any new weeds out of our country. We've already got enough. We don't need any more. So if it has to go into our biosecurity and our borders and not bringing new weeds in, or if it's only on one island and not the others, don't let it get spread to the other islands. When we have new weeds starting to emerge, we need to detect them early and eradicate them. We need to get in there and get rid of them when there's only a few. When the use of natural enemies becomes appropriate is when there is too much weed. It's not possible to control it, to eradicate it, or just stop it from spreading anymore. So we're not using natural enemies for when weeds first emerge and they're not very common yet. It's more appropriate to use other tools. And the SPREP team have a program where they can advise on that. wait for the weed to get worse. But yeah, respecting community view is essential. And we do absolutely have to respect our communities, and you know, things don't always go the way you want, but if you bide your time, you might sometimes succeed. Thank you for the great work you're doing at the moment with invasive weeds. I hope we have a similar program to deal with biocontrol agents for insects like little fire ants and GAS.
Giant African snail.
Giant African snail. Okay, I'm outside my area expertise there. I know the states had looked at natural enemies for red imported fire ants, and I think something quite dodgy was done around the giant African snails. I think something was released that was, again, a generalist that attacked other snails. So
yeah, there's a nematode.
Yeah, that's probably another whole seminar for another day with some experts in that area. I have a staff member who's working on natural enemies for vespular wasps, which is quite exciting. I think some of you might have some of those too. Has there been any case of people allergic to natural enemies introduced? None that I'm aware of.
No.
Okay, so that's the questions. Fantastic, thank you. We're going to move on now, and Michael's going to have a turn, and he's going to tell us about step 4, how we actually go about the process of getting permission to introduce new natural enemies.
Yeah, thanks, Andy. Okay, so up to now we have sought out what weed we're going to target. We've looked at what agents are out there or other control measures we can look at. We've chosen some agents and we've done some host testing. And now we've got to go through the step where we get approval to introduce a biological control agent or a natural enemy into a country. And often in many countries, it's exactly the same when you're introducing any other organism, whether it's fruit, vegetables, and things like that. There's this process called a import risk assessment. And natural enemies falls under the same category, and it's often done by your quarantine unit or the Department of Agriculture. And I'll go through the processes and what's required in one of these later. And if we've got time, I can show you one we did for other countries. And because when we introduce a natural enemy into a country, once it establishes, it's unlikely that it's going to be reversible. It's unlikely that we're going to be able to get rid of it. And so the decision to introduce them must be made carefully. And the science we use must be critical, and that's what we've talked about earlier, about the host testing to make sure it doesn't attack anything else. We need to weigh up the risks. There's always some, even though they might be very, very small, and the costs and the benefits. So the benefits of what we're going to do, of introducing our biocontrol agent, is that we have less weed, less impacts on biodiversity, and our farmers can increase productivity and things like that. But there is a bit of a cost. You know, these biocontrol or these natural enemy programs aren't cheap, as Lenny mentioned very early on, but the long-term benefits are great. And all these need to be weighed up in the IRA, or the import risk assessment. And because Lenny and I are closely involved in these from many countries, we also seek independent advice, because the regulators in these countries where we're going to introduce our natural enemy aren't necessarily sufficient in having done one of these before, or they're not necessarily proficient in weed biological control. And so they're relying on our expertise and our guidance. But we also think it's useful to have independent advice. And I'll go through some of the ways in which we can go down that path or to support applications. So we'll just go on to the next slide. And so these import risk assessments, this is where we provide as much information for the regulators to make a sound decision on whether the natural enemy can be imported. and it can be quite a lengthy document. So the information contains information on the target weed. So it includes the taxonomy of the target weed, which family this belongs to. But also attached to the taxonomy is, are there any closely related plants to that our weed found in the country? And this is because we want to know if our agent has been tested against those ones, or whether we need to test, do extra testing. And so understanding the taxonomy is great. So when the regulator is looking through the application, and then they look through what plants the insect has been tested against, they can say, oh, the insect has been tested against all our closely related species. Or if it hasn't, then that becomes a little red flag, and we might need to do further testing. We also look at the biology and the impacts of the plant. And again, this is because we want to know, we can relate, does the biocontrol agent likely to affect the plant? Is the biology of the insect going to merge with the biology of the plant, such as a seed feeder on a plant which only flowers once a year, or things like that? So we need to look at that. The impacts, this is quite critical, because if the impacts of the weed is not very great, why are we going to go to the trouble of releasing a biocontrol agent? But if the impacts are quite serious, then this justifies the release of a biological control agent. So impacts are really important. We also do the same thing in Australia, where we're not necessarily going to introduce natural enemies for weeds that have a local impact or a marginal impact. It's not worth the effort involved. And the control methods are important as well, because this is where we can relate biological control. Is the plant able to be controlled using other methods which may be considered safe or low risk? Or are the methods available that aren't suitable to be able to control the plant? So if you have a plant infecting waterways or paddocks, you aren't able to use feasible and cost-effective control methods, then biological control would serve a purpose in that regard. And so knowing what other options are available helps weigh up the risk or the low risk and the benefits of introducing a biological control agent. The other thing that we need to look at is the legislation of that weed. Is the plant a dedicated or registered as a species in the country, or is it just not even recognised? And so often the legislation will help dictate whether we go down a particular pathway of control or introducing a biocontrol agent. And as we mentioned on the first day, was we want to know, does the weed have any benefits? And the benefits could be medicinal, such as in mycania, or it could be useful for mulch and things like that, or shade, or it could be a tree species, like a garden plant, like African tulip tree. So we need to know what the benefits are and if there's going to be any conflicts of interest. And so documenting all of this into the import risk assessment allows the regulator to make an informed decision. Is pursuing biological control the best way and most effective way to proceed? So that gives a bit of a background on the target weed. So, excuse me. Now we put the information about our natural enemy. And this is where the things that we've talked about up to now are important. The biology of the insect, the host specificity testing, and Lindy was talked about that earlier. Does the insect feed on another plant? Or how much feeding does it do? Does it pre-produce on another plant? All these things need to be mentioned. And sometimes you might find that, because, and Lindy mentioned that, if you put an insect into a cage and you leave it there for a week on another plant, the insect gets very, very hungry after a few days, and it might take a nibble. So is that nibble important? Is it significant? Or is it just a nibble before it dies and it can't sustain itself? So those are the kind of things that get reported when we in our host testing, and then the regulator can see the results of this and whether it's significant or not. We also put there where the insect has been already released. And this is really critical, especially for the Pacific, because I had a message saying that they couldn't hear me, but
I can hear you, Michael.
Yeah, maybe there was just that one person.
If you can't hear, sometimes you need to go out of the Zoom call and come back in if the sound drops out.
Okay, thanks. And also what we'd like to document in our import risk assessment is where the insect has already been released and its effectiveness. And so for many agents that we've introduced into the Pacific in the past, they've had a long history of being released in other countries. So the 2 beetles on water hyacinth have been released in over 35 countries globally, and the effectiveness is very high. And the testing has also been thorough before it's been introduced. And so we put all that information in there, and that gives the regulator a lot more confidence that the insect not only has been thoroughly tested, but it's been tried in all these countries, doesn't attack anything else, and is a highly useful insect. And if you look at that compared to, say, one of the insects I referred to on Lantana, which doesn't do much damage, well, you wouldn't necessarily put in release an insect that doesn't do damage in another country. You'd think, well, if it doesn't do damage in one country, is it likely to do damage in ours? And so you might just consider whether that's an appropriate method or an appropriate agent to introduce. Lindy mentioned that some of the agents which are found in some Pacific countries are not found in others, and that also gives people a bit of strength. So there's agents present in Fiji, Vanuatu, Cook Islands, Papua New Guinea, which aren't present in many of the other countries in the Pacific. And so those countries who are interested in introducing natural enemies can seek guidance or seek information from those other countries. And Lindy mentioned we're now a nice little regional hub where we can swap ideas. So all this information gets also put into the import risk assessment. The other thing I mentioned before, that the risk assessment should be have independent input. And some of the independent input could be through published papers or scientific papers. And this is because scientific papers are peer reviewed. When we submit a paper for publication in a journal, it goes out to some people that we've never met before, where they're anonymous. They'll look at the paper, they'll look at the science, they'll look at the conclusions, and state whether it's bona fide or whether it's rigorous or scientifically sound. And if it's not, they'll let us know. But if it is, the paper will then get it published. So that shows that the testing procedure was sound and safe, and the conclusions that we have back up, or sorry, the conclusions that we make are supported by the data. And so the document itself can end up being about 20 or 30 pages long, and it's all in little sections. So even though there's a template, even though it looks complicated, there's a template with all these questions and we just have to fill them in. So I don't know, we've only got 8 minutes before we go.
I would suggest, Michael, what we should do is put on the shared drive some examples.
Yeah, I was going to suggest that.
Look at some real life examples of it.
Yeah, I was going to suggest that. So because I don't have time to go through one, I've suggested that we'll put the one that we prepared for the mimosa psyllid for Vanuatu. We'll put that up on the shared drive and you can have a look at just what goes into them. The information on the insect is often the same, whether it's for one country or another, but the information for the country is obviously country specific. And we've actually sent a new one to Haggard to look at for Niue. So he's going to look through it all and see if he thinks it's suitable. And I think that's, I don't think there's anything else.
Thanks, Mike. What we're going to do now is again hear from you guys, and so we'd like to know, do you think it's going to be easy or hard to get permission to release natural enemies in your country? So you could put a yes or an easy or a hard, very hard, impossible. Type in your responses in Menti. I've got not easy, hard. And if you think it is going to be difficult, are you able to tell us why? So we've got one easy, another not easy.
I think the other thing to
Sure.
is that for the very first few applications is that Linear or I will certainly assist in writing them, as we've done for other countries. So don't feel scared about writing them. We're happy to help you with them. And it's just a matter of whether the processes in your country is amenable to introducing natural enemies.
And I think too, if it's something that your country hasn't done for a while, we have to expect that the first time might be more difficult because everybody's getting familiar with the process and the legislation and what's required, and it will get easier from there. But what we have found is it usually is possible with the right information and the right support to get permission, but it can take some time. So thank you for all those answers. Are there any, oh, thank you, someone's put, there's a stigma on lens from past experiences. Yes, and that's why we must always explain to people that when we're working with weeds and natural enemies, it's completely different to the generalist mistakes that were made in the past. Are there any questions that anybody would like to submit now around the whole import risk assessment or anything else we've covered today? Okay, here's some more feedback. Thank you, Hard, but the plant has significant value to the community. Yeah. Yeah, that's right. If everybody agrees it's a really bad weed, it's easier. If there's some conflicts of interest, and that's where we talked yesterday, sometimes we need to measure the value, do some economic cost benefit type analysis. Can another country use, well, yes, we've had some sharing of this. So if one country has done an RIA, then often there's very little needs to be done to tailor it for another country unless there's any new additional information. So again, this is where if we can all work together as a network across the Pacific, we can have efficiencies and we can support and help each other to make this easier and cheaper and more feasible. Just on that, often we just edit a previous IRA, but in the case of Tonga, I actually sent them an IRA I prepared for Vanuatu and asked them if the information in this IRA was suitable for them, and I was prepared to just rewrite it for Tonga, and they actually wrote back and said, yes, we'll issue you a permit. So that didn't even require any changes. They just accepted it as it was. So just depends on the country. Okay, so what role will the exporting country play in an IRA? That's a good question. If it's one Pacific country sharing with another, I guess they will be providing information to help inform the IRA. We do have, when we're sometimes dealing with new natural enemies not available in the Pacific, we have to work with the country for which that is native biodiversity, and whether or not they will give us a permit to allow us to use as a natural enemy. And sometimes that can be quite a long process. Some countries are less willing to share their biodiversity, even though this is for public good. So the role that an exporting country will play can vary a lot depending on the context. Sources of information from Google and Cabby. Are there other places, sources where we can get reliable sources for compiling an IRA? Michael, do you want to answer that question?
Yeah, look, a lot of information is on, we've already got information for those target weeds anyway. So when we're looking at, say, importing an agent from, say, which is found in Fiji, and we're going to introduce it into Samoa or American Samoa or some other country, the information on those plants and their agent is already well documented. Where we've got a new target, then that becomes a little bit more complicated because the information is in hand, but we can get it from Cabby. They've got that invasive species compendium, a very good source. Countries should have or may have information about the target weed in their own country, because often the IRA refers to what are the impacts in their country as well as others. But again, I think have a look at the IRA that I'm going to share with you after this session is finished. And people can feel free to email me specific questions. And just remember that when we're doing our first IRA for your particular country, Lindy and I will bound to be involved and help you through it, as we've done for other countries as well. So it's not as though we're going to throw you in the deep end.
Have you experienced land tenure issues while facilitating NENS communities versus private land? I guess the thing is that once a natural enemy has been released in a country, it will go everywhere, regardless of who owns what land. And so this decision about whether or not to introduce and release natural enemy has to be carefully considered, because it's too late once you've actually brought it into the country. You can't keep it on some land and not on others. Thanks for those questions. Again, we just want to check that you're able to follow what we're saying. We're not speaking too fast, not using too many big words without explaining them. That's fantastic. Great. That's very reassuring. Thank you. What we will do is let you provide some feedback on the session or any suggestions for the next two sessions. We will review that after the session's finished. So thank you if you have any thing you want to tell us there. We are just going to look at the homework. If you're going to be involved in NENS in your country, you're going to need to be able to talk to people and your communities about NENS. You need to have practice so that you can say very confidently to someone why it's safe. If you've got someone who's concerned, how are you going to answer that question? How are you going to tell them about the benefits? But also that you're able to be upfront and honest about the drawbacks, that the use of natural enemies is not going to be fast. It may not provide the level of control we'd hoped for. The agents might not even establish. Also want you to have a think about what experts and resources that you have within your countries, because there will be questions about the flora and fauna of your countries that will need to be addressed as part of NEN's work. Who's going to help us to find plants if we need to find them to do host testing, for example. If you have a native plant, we need to test who's going to help, who can assist with that sort of level of detail of information. So that's your homework. But again, all these people on the screen, if we can all work together and all help each other, we can beat some of these invasive weeds. So that ends the session for today. You can go away and practice your homework. And we look forward to seeing you again tomorrow. We're going to talk about rearing and release and assessment and monitoring and evaluation. And we look forward to seeing you again tomorrow. Ka kite anō.
Right.